Hi everyone, welcome to the video describing how to use the deposition system in the Natural Products Atlas. The deposition system is the only component of the Atlas website which requires you to have a login. This is to prevent malicious attacks of the website. So if you don't have a login yet for the Atlas, you can do so. This button up here in the top right will say login, and if you click it, it will give you a simple form which you can use to get a login established. Occasionally people run into difficulties with the validation email being lost in their junk folders or being filtered by the university. If that's true, then just send an email to support@npatlas.org and we will sort that out for you behind the scenes. So we're keen to get you up and running as easily as possible. So this deposition system is really for articles that are not already in the Atlas. So if you have found an example of a molecule that's missing from a paper that already has some molecules included, we would prefer you to use the additions and corrections section, and there will be a separate web tutorial on that. The Atlas always records the original isolation of compounds, so the deposition system should be used for new compounds only. If you have a study where you report both new and known chemistry in the same paper and you wish to add all those compounds, then we ask that you use one deposition with the current paper for all the new molecules, and you then find from the literature the original isolation papers for the known molecules and deposit them as separate articles. Consequently, we don't accept depositions of data from review articles. And currently we don't have the capacity to handle patent literature either, although we are working on trying to resolve that. So the Natural Products Atlas, as a reminder, it's for microbial compounds only. So this means that we don't accept depositions from either plant or marine invertebrate sources. However, if a molecule was originally described as a plant or an invertebrate derived molecule and was subsequently found to come from a microorganism as well, then that's a legitimate deposition. And in those cases, we'd ask that you deposit the first instance of microbial isolation in order to keep things consistent. So today we're going to use one of our own papers as an example. So we're going to work through depositing data from this article here from 2015, describing the discovery of a family of phosphorus-containing natural products called the Selenopostins. So if we wanted to deposit this article into the Atlas, we would start by going here to the deposit page. This button is on every page of the site. And what you'll see here is that the number of requirements for deposition is deliberately kept very low. So we've tried to restrict this to just the bare minimum information we need so that deposition is very straightforward and that people can do it expediently. So we only need 4 pieces of information. We need the information about the citation, preferably the DOI. We'll come back to that in a minute. We need the names of each of the compounds you wish to deposit and their corresponding structures. And then we need information about the taxonomy of the producing organism as well. So we'll work through these from the top. So if we start with the DOI, almost every journal article now has a DOI assigned to it. And the DOI is a very useful way for us to obtain all the other citation information, like the name of the journal and the page numbers and all the other details. So the DOI is a unique identifier for each article. If we go back to our Selenopostin page, what you'll find is that about half of the publishers display the DOI for each article on the article page somewhere near the top, usually just below the title somewhere. And in this case, in the ACS web environment, they're always here. So the easiest way to copy this is to Control-click or right-click on the Mac, on Windows, and then copy link location here. And if we then go back to the deposit page and go into the DOI box and paste that, you'll see the DOI is deposited here. You'll see that because I'm working at home at the moment, it is preceded by the proxy information from my library. Don't worry about that. Our system will strip that information when we import the data. We're only interested in this piece of the number that comes. That's not very good. Let's try again. We're only interested in this part of the number here, which comes afterwards. If you prefer, you can strip this information at the front and relieve just the numbers. So if you would rather do that, that's totally fine. So the other half of the publishers put the DOI in a rather unusual place. So here we have an article from the Journal of Antibiotics. So the Nature journals do this, and a number of other big publishing houses do similar things. So in these cases, the DOI isn't displayed in the top header here, but rather is in a box at the bottom of the page called About This Article. So if we follow this About This Article link down to the bottom, what we find is that you get the information about the citation, and then again you get the DOI down here. And you can do the same thing, right-click, copy link location. Okay, so the next thing we need is the names of the compounds, so we can just put the first one in here. There are lots in this article. I'm not going to do them all, but we'll do a few just to make that clear. If you only have one or two compounds to add and these other boxes are empty, that's fine. If you have more than 3 compounds to add, you can use the plus button here, and that will add additional rows to this form. So we could add a couple more here just to make the point. We also need the structural information for each compound. So the best places to get the structures are either from PubChem if you have the original files, or from, either from ChemDraw if you have the original files, or from PubChem if you don't have the structure in hand. So if you have the original structures in ChemDraw, then you can select each structure and then go to the top menu bar and choose Edit, Copy As, Smiles, and that will copy the smile string, and you can just paste that into the box here. We do ask that you do them one at a time, so please don't copy structures that have R groups or other ambiguous features. We need a unique molecule in each row of the page here. Alternatively, if you don't have the structure, the most reliable place to get those is PubChem. So if we go to PubChem here, if I put in selenopostin A, we can search that molecule, and we find that that does come up. It's important to keep an eye on the synonyms that they list here. Occasionally there are corruptions, and wrong compound name will be listed as a synonym as well, so you've just got to watch out to make sure you're picking the right one. So in this case, we're going to choose selenopostin A. We'll go to the selenopostin page, and the first thing you see then is the structure. It's definitely worth looking at the structure to make sure it's accurate. In general, PubChem is excellent, but of course there are sometimes problems or unexpected features, so it's worth having a look. So in order to get the structure from PubChem, the simplest thing to do is to scroll down here to section 2, which are the names and identifiers, and then down here you're going to see IUPAC name, INCHI key, and then canonical smiles and isomeric smiles. So if a compound has chiral centers, and if those chiral centers are defined, then there will be an isomeric smiles entry. And this is the preferred entry, because it includes the configurational information. If the molecule is planar, or if the configurations have not been assigned, then there will only be a canonical smiles entry. So when you choose smiles strings from PubChem, the preference is to select and copy the isomeric smiles. The alternative is to select and copy the canonical smiles as a second choice. So I'm just going to copy this one here, and I'm going to go back to my deposit page. I'm going to deposit that smiles like this, and it should look like this. It should be mostly capital C's and some ats and brackets and things like that, and then you know things have gone correctly. So I'm not going to do the others for the sake of time here, but we would if we were going to submit the serial, of course. So then the last thing is to select the type of organism. So we have split these only into these 2 very general classes. So you choose the appropriate one. In our case, this was a bacterial isolation project. And then the genus, Selenospora, and the species, in this case, SP dot, or if you know the species, you should add it. If you know the species and it has an ATCC number, it's okay to put the ATCC number at the end of the species name as well. It is very helpful for us if this information is clearly separated like this. It makes the importing a lot quicker. If you don't know, you can write unknown in the genus box. But if you don't know that whether or not it is truly microbial, then obviously it's not an appropriate compound for deposition. So then the last thing you would do is press the submit button, and you should get a pop-up window thanking you for your deposition. And you should also get an email which provides you with the information about what you deposited. All of these depositions then go into a holding pen for the Atlas team to do final cross-checks and validation to make sure that all the data are standardized before they get inserted into the Atlas. And you will see your depositions appear in the next version of the database which is released. And typically we try and do those once a quarter. So that's all there is to deposition. We definitely thank you for your contribution, and we look forward to seeing your molecules join the bank of information available to the natural products community.